Antes que não dá, tá?
Hey dudes, how we all going? Welcome back, guys. Great turnout again. Awesome job. Doing well, Luke, yourself? Yes, doing very well. Today is my birthday. 26 today. Oh, geez. Thanks, guys. Cheers. Yeah. Yeah, have a good day. Hope you guys are all doing as well. I know we mentioned last week, it's not the most interesting tutorial this week, but it's whatever. Look, we're gonna spice things up a little. We're gonna pretend we're having a little birthday party, and it's gonna be fun. We're gonna play some games, like labeling the brain. You know. Let's get this up. Stack it and let's go for a drink. Would love to agree with you. I probably shouldn't. All right, I reckon we're ready to get started. Should scull a beer or something. Nah. Maybe not. Might lose my job. All right. Hopefully, yeah, I am sure it's getting this. So hopefully you can all see the slides today. I made these myself, by the way. I'm assuming these are like way better than what you're used to. I put a lot of effort into these slides. Look at these gradients. It's like light blue, And then you got this green, very clean. So what we're, yeah, we are up to week 2. So you should have already had, oh, sorry, week 3. So you guys should have just come from your lecture with David, yeah? Synaptic transition. As I mentioned, it's a bit of a rough start, getting straight into like pretty hardcore neurochemistry. Like it's no joke, that kind of stuff. So I will continue on with synaptic transition and membrane potentials and stuff like that next week. So this week we're just looking at, as you can see, August 13, we're just gonna be doing more like nervous system stuff. So I think based on some stuff that I saw that Taryn posted, you guys have already had to do like some anatomy stuff, like some basic tutorials. So I apologize if some of this seems like revision to you, but yeah, we're gonna get straight into it. So for any of you that happened to print off the prac manual, like Tanner and I don't really recommend it. We kind of made these slides so that they would be like all-encompassing. So you can look at these slides and you don't need to worry about like the lab manual at all. If you did happen to print them off though, or you're interested in printing them off, it is kind of a good idea because it contains all the diagrams. So and you can be filling out these diagrams as you go along. That's not necessary to do well in the subject. A lot of the like anatomy focus you will do hands-on. So you have, I believe you do 4 of the face-to-face anatomy pracs throughout the semester. And that's when you'll really get into like you'll get to see actual cadavers and you'll get to see where like nerves and features of the brain are in real life. Whereas these are just diagrams, they're not, you know, like super engaging, and it's not as easy to sort of like understand where everything is. But yeah, if it's something that you want to be like, you want to take like furious notes, yeah, absolutely, you can refer to the lab manual as well. Okay, so starting off with gross features of the brain, we have our olfactory bulb at number 1. And number 2 is the cerebrum. So most of this picture is referring to the cerebrum or your cerebral cortex. It's basically just like the brain, like it's where all your main functions are. And the olfactory bulb is just one of those features that is just, for some reason, it's one of these like specific organs that is on the outside of the brain. Whereas most of it, yeah, it's just cortex. And then you have the back of it is the cerebellum, and then you have your brainstem. But mostly, when you talk about gross features of the brain, what you're mostly talking about is your cerebrum. All right, so looking at your gyri, sulci, and your fissures. So your gyri, so I'll start with your sulci. So your sulci are your grooves. They're like the like fissures or like grooves in the brain. So if you think about the word sulci, it's kind of like to sulk, and sulking is where you make like a sad face, like a I don't know how else to describe it other than that sound effect. But so to sulk means like the groove, the actual like inside bit, and your gyri is like the top round surface of the brain. So if you think of the brain as like spaghetti on a plate, the actual like noodles are your gyri, and then like the gaps in between the noodles, that's your sulci. And the reason that we have these is that it increases the surface area of the brain, which was really important for evolution. Okay, so and then this diagram is just explaining. Oh, and a fissure is just a really deep sulci. So your lateral sulci is the main sulci that separates your temporal lobe, which is just underneath 5, from your frontal and your parietal lobes, which is just above it. And then 6, 7, and 8 are focused around your central sulcus. So again, your sulci are your like grooves, and then on either side of those you have your gyri. So you have central, and then you have your precentral gyrus and your postcentral gyrus. That should make that's pretty straightforward. And then you have your superior temporal gyrus as well. Superior meaning like on top, and temporal obviously refers to your temporal lobe. All right, so then you have your cerebral lobes and the insula. Starting with the insula, you can see on the diagram on the right, the diagram's just showing these like metal prongs pulling apart the brain, and it's exposing the insula underneath, and it is involved in taste. This is just the four main regions of the cortex. So your temporal lobe, which is involved in like auditory functions, language recognition. You have your frontal lobe, which is important for just like so many things, but I guess generally it's involved in like what makes us human. So like decision making, higher order cognition, and some memory stuff as well. Memory is in a lot of different places as well. Number 3, parietal lobe, is your involved in like sensation, as well as some like sensory movement stuff. And then 4 is your occipital lobe, obviously heavily involved in vision. Then we have your major sensory, motor, and association areas. So these are just talking about like the differences between like your general cerebral cortex and then breaking it down into like really specific cortexes or cortices. So number 6, you can see your auditory cortex is located in your temporal lobe. So think of a lobe as like a really broad, like really general region, and a cortex is like an area that serves a specific function. So your auditory cortex, obviously involved in hearing. Number 7 is your inferotemporal cortex. So the infero here just refers to like below the temporal cortex, or like at the bottom of it. 8 is your prefrontal cortex. This is really important as well as an area of the frontal lobe. 9 is your premotor area. And 10 is your supplementary motor cortex. Moving on to 11, you have your primary motor cortex. 12, you have your somatosensory cortex. So somatosensation is really about, it's like a complex mix of sensation and reactions and that kind of thing. Really important in movement and reactions particularly. 13 is your posterior parietal cortex. 14 is your visual cortex. Obviously it's involved in vision. And 15 is your gustatory cortex. So I think we saw a diagram, yeah, just before, where that exact same diagram was exposing the insula. And so in this case, the insula and the gustatory cortex are referring to sort of the same area, but it's all involved in taste. One second. Right, now looking at the medial surface of the brain. So if you take a cross section, cut someone's brain in half, this is what a medial cross section would look. So you have your medulla at number 1, 2 is your pons, number 5 is your midbrain region, but it is made up of your tectum and your tegmentum. 6 is your hypothalamus. You guys would know, or would have heard all about your hypothalamus in a bunch of other different subjects. It's just involved in so many different things. 7 is your thalamus, 8 is your pineal body, and 9 is your cerebellum. So cerebellum is actually referring to quite a large organ here in this diagram. So if you guys are wondering kind of at this point, like how much of this do you really need to know? I guess it will depend on, like later on, we're going to look at neurotransmitters and like where each of them specifically are produced and what they interact with. So you will see a lot of overlap. It's not just like we do this one sort of tutorial on anatomy and then we never come back to it and you're just like, okay, well, was I supposed to like memorize all those different regions? Not necessarily. I guess technically we could just in an exam just have a diagram and be like, okay, where's the tegmentum? It's unlikely. We're more likely to ask you questions that would only really require your knowledge of anatomy if you were to say, talk about where like what this organ, like hypothalamus, produces certain hormones, and how is that important to, like, how is its location relevant to the way that it communicates with other organs of the body. It's kind of hard to like explain this, having not done the tutorial on the diffuse modulatory systems. So it will make a bit more sense when we get into that, because that brings back a lot more anatomy as well. So just bear with us this week, guys, because it's just a lot of anatomy. But I mean, it's neuroscience. It's not the worst kind of anatomy, to be honest. Right. So we have the 4 brain structures. Number 10 is your optic chiasm. So you actually come back to this a couple of different times later on as well. So your optic chiasm is where your left and right visual nerves cross over, which is an important thing. 11 is your olfactory bulb. So this is involved in smell. And weirdly, it's just at the very sort of tip of your brain, and it's outside of the cortex. Kind of a weird spot for it. But interestingly, it is like one of the shortest nerve pathways in the body to go straight from the nose to the olfactory bulb. It's really, really short. And in some cases, your sense of smell will bypass. Like the thing is that your central areas of the brain are kind of thought of as your like post office. So they receive all sensory information from the body, and then they send it to the specific areas of the brain where they're meant to be processed. So in the case of like seeing something, for example, passes along your ocular, your visual nerve to your brain, so the central areas of the brain, and then it gets sent to the occipital lobe for higher order processing. And just in the case of like olfaction or smelling, it's just more of a direct pathway. What do we get up to? 12 is your fornix, 13 is the cingulate cortex, 14 is your corpus callosum. So this, I think there's a better diagram of this coming up actually. Your corpus callosum is one of the main structures that connects the left and right hemispheres of the brain. And it's kind of important in just sort of communicating information between the 2 different hemispheres. And then number 15 is your calcarine fissure. Yeah, so the thalamus, before I go past that, did we talk about the thalamus? Yeah, we did at number 7. So the thalamus is the Oz post of the brain. It's what like receives all the signals and then sends them to their like higher order processing. Just something I wanted to mention. And then finally, you have your hippocampus, strongly, and amygdala, which are strongly involved in like emotional regulation and memory, which are themselves highly associated. But we'll be coming back a lot to both of these in particular. Like we get to do heaps of stuff on memory, and we do like anxiety and depression as well. So these 2 both come up again as well. And we do like stress and all that kind of stuff. Stress and depression and anxiety, that kind of thing. Okay, then we have your ventricular system. So your ventricles are what produce and store your cerebrospinal fluid. So this is like, it's kind of like a special kind of blood. It is responsible for like kind of nourishing and just moving fluids throughout your brain and spinal cord, so your central nervous system. So 1 is your spinal column. 2 is your 4th ventricle, so you have 4 ventricles. 3 is your cerebral aqueduct, which is just connecting your 4th ventricle to your 3rd ventricle. And then on this diagram on the right, you can see your lateral ventricles. So you have 1 on each side, so your 1st and your 2nd. So 4 ventricles, and this is basically your main like ventricular system. Okay, ventral surface of the brain. So ventral in this context means like the underside of the brain. So at 6, you have your medulla, or like the underside of it. 7 is pointing to like 7 different things. It's actually indicating, if you can see these sort of noodly end strands, it's pointing to your cranial nerves. And they're actually cut off in this one, and it's only pointing to 7 of them. So you actually have 12 cranial nerves, which we'll go through in a couple of slides' time. And they're really, really important. So you'll actually get to learn all of them, which is great, all 12 of them. 8 is your optic nerve. It is one of your cranial nerves. 9 is your olfactory bulb. So as we like looked at before, we had a sort of side view of the brain, and you can see the olfactory bulb was just pointing down the bottom of the brain. So it sort of sticks out of your cortex. 10 to optic chiasm, so this is just another view of it, and you can see that it forms that cross shape. So that is literally your two optic nerves crossing over each other as they head into the brain for higher processing. 11 is your optic tract, 12 is your hypothalamus, 13 mammillary bodies, 14 and 15 are your midbrain and your pons. Now looking at the dorsal surface of the brain. So dorsal is like a dorsal fin. You're like on a dolphin, a dorsal fin is the one on the back. So looking at the top of the head. So you have your longitudinal fissure. So again, fissure is just a really like big sulci. And it is the biggest fissure that you have in the brain. And it separates your left hemisphere from your right hemisphere. 2 is your central sulcus. We saw that in like one of the first diagrams. 3 and 4 are your left and right hemispheres, and then 5 is your corpus callosum. So again, you can see in this diagram, it's using these like middle prongs to pry apart the hemispheres. And if you were able to do that, looking down, you would see the corpus callosum. And you can, yeah, literally do that. If you had someone's brain, you could pry apart the left and right hemispheres. They're not actually fused. And the corpus callosum would be one of the main sort of bodies of tissue that is actually connecting the brain together. This is continuing on the dorsal surface of the brain. It's just removed the cerebral cortex. So you actually have two hemispheres of your cerebellum. So you have your left and right. 7 is your vermis and 9 is your spinal cord. So your spinal cord attaches to the sort of bottom of the cerebellum. This is another diagram where the cerebrum and cerebellum are both removed. So it's mainly just looking at like the midbrain and the pons and that kind of thing. So 10 is your pons, 11 is your midbrain. Then you have your thalamus. 13, 14, and 15 are your pineal body, superior colliculus, and inferior colliculus. Do I want to say anything about that? The superior colliculus is like a multi-sensory structure, and the inferior colliculus is just the midbrain nucleus of the auditory pathway. So I wouldn't be super, I wouldn't be super interested in either of those. 16 is your cerebellar peduncle, and 17 is the floor of the 14th ventricle. So we're just trying to basically give you different angles of the brain. This is all like really good for, yeah, I'll say this again because it is important, but it's really good for later on in the semester when you know, we'll have a random tutorial and I'll mention like, oh, this is produced by the pineal body. And you'll be like, oh, where was the pineal body? Or I'll bring up something about the ventricles and you'll be like, oh, I can't remember where they were. So you can come back to these slides. They're just a really good source of information where everything is. Not super necessary for you to be like, basically writing all this down somewhere. That's kind of what these slides and these recordings are for. All right, then you have spinal cord. Yeah, so this is going to seem pretty overwhelming. Do bear in mind though that I'm almost pretty sure that you have a one of your anatomy pracs will be on spinal cord. And you will get to do this a little bit better. So this is just going to be a brief sort of introduction into spinal cord anatomy. These first 4 are probably the most important ones that I want you to take away from today. So you have your dura mater, your arachnoid, and then you have your pia mater. And in between your arachnoid and your pia mater is a space called the subarachnoid space. So the subarachnoid space isn't actually like a thing, it's more just an area. Your actual 3 layers are your dura mater, your arachnoid, and your pia mater. Those are like your 3 those are your 3 layers of the meninges. So they protect your brain and spinal cord. They protect your central nervous system. So that's 1, 2, 3, and 4. 5 is looking at your ventral column. 6 is your ventral horn, and 7, 8, and 9 are looking at your lateral column, your dorsal horn, and your dorsal column. And then you've got these two arrows here that are indicating where sort of ventral and dorsal are in relation to each other. Moving on, at 10 you have your spinal canal, then your lateral horn. Then you have your dorsal root filaments, your dorsal root, and your dorsal root ganglion. "15 is your spinal nerve, 16 is your ventral root, and 17 are your ventral root filaments. What is the purpose of each of the three? Of the dura mater, arachnoid, and pia mater? They're just mainly like protective. So they would serve like as a physical protection. We haven't quite talked about yet the blood-brain barrier and that kind of thing, but I guess it's important. All I'll say for now is that the central nervous system is a little bit different from the rest of the body in the way that it protects itself from the rest of the body, kind of. Like the central nervous system knows that it's better than everything else, that it's more important, so it has special exceptions to the way that blood is able to enter the central nervous system. And yeah, I will say about the meninges that it is just, again, a special case where the central nervous system has evolved not necessarily better structures, but just different structures because of the unique way that neurons function compared to just normal cells. Like muscle cells and that kind of thing. So that might become a bit clearer as things go on. I think we talk about the blood-brain barrier, maybe not next week, when we do neurotransmitters and the diffuse modulatory systems. Okay, cranial nerves, awesome, we're here. Very cool slide. Normally, you would also have a face-to-face anatomy on cranial nerves. I'm hoping you guys still get to do that because this is a really, really important topic. And yeah, basically everything on this slide is something that you will be expected to know in this subject. So from 1 to 12 in Roman numerals, because that's just how cranial nerves are lettered. From 1 to 12, you have your 12 cranial nerves. And in brackets, I've put whether they are sensory, so whether they are receiving sensory information from your eyes, your ears, or sensation in your fingers, or whether they are motor, so whether they are picking responsible for reflexes or for voluntary movement, or just anything in general that involves telling a muscle to do something. So for example, oculomotor is involved in moving your eye muscles. So it's a motor nerve. And then you have some trigeminal, which are involved in both sensory and motor. So I've just written both. You guys need to know so first of all, you need to know all 12 cranial nerves. You need to know their numbers. You need to know where they are, which is a really important one. So you can see on the diagram on the right, they are numbered conveniently from top to bottom, from the front of the brain downwards, if you're looking at a ventral surface of the brain, which is pretty convenient. That might be an easy way to remember it. What else? The echolocation, where they are. And yeah, and you need to know if they're sensory, motor, or both. Yeah, that's also really important. And I would find it just generally helpful to sort of, just do a little bit of research on each one. Just quick Google search, what is the vagus nerve? Vagus nerve is really, really important. It's just involved in so many things. And we used to say that if you had a question and they were oh, what nerve does this? And you really weren't sure, you can just guess vagus because it's involved in so many different things. But yeah, so don't worry too much about you having to come back to this and absolutely getting it right quickly. You do get to revisit these in Anatomy Practical 4, actually. Yeah, so that's gonna be really good for you guys. But I'll go through them all. So you have your olfactory, optic, oculomotor, your trochlear, trigeminal, and your abducens. Then you have your facial nerve, your auditory vestibular nerve, your glossopharyngeal, your vagus, your spinal accessory, and your hypoglossal nerve. All right. Let's keep moving. Oh, awesome. We are out of the anatomy. Awesome. That's so good. I promise you the rest of the semester is not like this. I mean, the anatomy stuff that you do at UTS, if you do end up coming in to do it, again, it's not compulsory, but if you do come in to do the face-to-face anatomy stuff, which I highly recommend you do, because it's really cool, you get to touch cadavers and stuff, that is obviously going to be really anatomy focused, but for the most part, that was kind of it. We're mostly just going to be doing, yeah, like case studies. We do a bunch of case studies throughout. And yeah, we do a lot of content that like complements the lectures that your lecturers are going to be doing. So this is kind of a weird case study, but we do it because it's a way of us being able to be like, okay, now we can talk about like MRI and fMRI and that kind of thing. And it even talks a little bit about ventricles, which we just talked about. So just to quickly read through the case study. So it's about Mr. RW, whose name has been redacted. He has clinical history of an atypical gait with brisk lower limb reflexes and incontinence. He was being treated for Parkinson's with L-DOPA. So L-DOPA is an analog of dopamine. I don't really want to talk about this yet. That's going to be for when we do neurotransmitters. Yeah, we'll push forward. Over the next 3 to 6 months, his health rapidly declines and he was no longer able to drive. His local doctor assumed he had a stroke and added warfarin to his treatment. Yep. Walking initiation, movements became increasingly difficult. Memory loss became a problem. Okay, so they did an MRI scan and they diagnosed normal pressure hydrocephalus. So it is usually due to a gradual blockage of the cerebrospinal fluid. So as I mentioned before, your ventricular system is the main carrier of your cerebrospinal fluid. And this is indicating that a blockage there is causing enlargement of his ventricles, which I'm going to show in just a second. So NPH is an unusual cause of dementia, so that's why he was having memory loss. And it can occur as a complication of brain infection. In patients with normal pressure hydrocephalus, although the ventricles enlarge, the pressure of the cerebrospinal fluid remains within normal range. So the gist of it is just that he's having drainage problems. So he's having problems disposing of or moving his cerebrospinal fluid around, and it is causing the spaces which hold that fluid to engorge. And for various reasons, that is incredibly bad. If you have fluid buildup, or if you have infection, or you just have general swelling in the brain, it is really, really bad, especially if it's a chronic condition, because the brain will literally just shrink. So your cerebral cortex, which is where like your like really like memory is involved in there. A lot of your like just general essential functions are involved there. Those will just start to shrink at the expense of like expanding fluid, which is a bit rough. Obviously, that's causing dementia, and this guy's having all kinds of problems. Characterized by dementia, incontinence, symptoms progressively worsen over weeks, So his treatment. So treatment of normal pressure hydrocephalus is tricky, improvement is never guaranteed, and trying to drain the cerebrospinal fluid is fraught with risks. Again, because of the complex way in which our nervous systems have built themselves and sort of the protective barriers that they've put up to protect itself, can make it difficult to do things like surgery, and even just like treat people with pharmaceuticals. Again, as we'll talk about more when we do like the blood-brain barrier and neurotransmission, can be really tricky to get like drugs into people's central nervous system. Okay, and we'll undergo a further CAT scan to manage the shunt flow. Did it mention? Yeah, so they put a magnetic shunt in to drain his cerebrospinal fluid. And because of that, he experienced an immediate improvement in gait and memory. Awesome. Good to hear. Okay, so this is just Yeah. Okay. So a lot of these questions that you'll see in the in my slides are literally straight from the tutorial. So because we can take questions like exam questions, because we can take them from the tutorial manual, that's why I made sure that they would all be in tutorial slides. So you can, if you choose, to just completely ignore the tutorial slides, like never even look at them, not tutorial slides, don't ignore my tutorial slides, completely ignore the manual. I don't even know if any of you have like even opened it, but you can completely ignore it if you want. And because all the questions, all the multiple choice questions, all the like, as well as the answers to them are all in these slides. So that's just why you'll see randomly I'll have questions in here like compare this and list the obvious differences. It's just part of the manual, and it's what we've always done for neuroscience. Like we've always gone through these questions together. So on your left here is diagram that was taken from the lab manual, and I provided a healthy scan for comparison. So this dark region, which hopefully you can all see, is his lateral ventricles. And they should not look that big. They are way too big. This is what this sort of light area is more so what your ventricles need to look like. So the fact that they are that engorged is a bit of a worry because of all this cerebrum that is being shrunk as a result of his ventricles enlarging. And here's just another diagram of it. So these white areas on the right are what your ventricular system is meant to look like. So they're white because they contain cerebrospinal fluid, and this gray matter is indicating your cortex. And on the left, I know the color is a little bit different, so I'll just explain that. The one on the left, it's the gray that is his ventricles. So that's all fluid. Yeah, not great. So if you were to list the obvious differences, this is sort of what we would expect you to be able to list. So enlargement, dilation of the ventricles. The lateral ventricles appear as a single ventricle. So the sort of anatomical feature that separates them isn't evident. Thinning of the ventricle walls, that's also true. So good analogy of this is when you have a balloon and you haven't inflated it, the like skin or the walls of the balloon seem quite thick. And then when you blow it up to maximum capacity, the walls of the balloon now seem a lot thinner. So it's the exact same thing that's happening here, is that the ventricles are a balloon and they're being blown up with fluid, and so their walls are getting thinner. So it's important that you recognize that as these ventricles enlarge, their walls also thin. Decreased diameter of the cerebral cortex, yeah. Your head is contained by a skull. You know, it's sort of the only region in the body where you have important structures that are fully enclosed in like a bone. It's not a bone cage, it's a bone box. So yeah, I should have mentioned this before, but that's basically why you won't see like people's heads swell up with like normal pressure hydrocephalus. It's just that there is swelling occurring, but it is happening at the expense of important brain tissue. And the final dot point is just the diencephalon and the brainstem are no longer visible in the MRI due to the ventricular dilation. Cool. What is this? Yeah, so there's a bunch of sort of material in your lab manuals related to MRI, so we'll just read through that quickly, and also a little bit on positron emission topography, or PET. So it's used to monitor brain activity while a subject performs a task. So PET is the one where like the areas of the brain will like actually light up when they're being activated. So if you're performing like a language task or something, you might see language areas in the temporal region light up on a PET scan. That's all it really mentions. It's mostly concerned about with MRI. So MRI is important tool for brain and cognitive research. That's important. The key to MRI is making the protons in the atom jump from one state to the other, high energy versus low energy. This is getting quite into physics. Energy is added to the protons by passing an electromagnetic wave through the body while it is positioned between the poles of a large magnet, which is why an MRI is basically a large magnet. 3D images can be produced in about 15 minutes, which is really good. That's really quick. And there's clear differentiation between white and gray matter, which is quite important for diagnostics. And it is possible to see the effects of demyelinating diseases and lesions in the brain. So lesions are something that are very largely studied in neuroscience. They're really important. For example, you see lesions in like the hypothalamus, and then we like to study like what specific functions break down as a result of that. Why is Mr. RW not being referred to for another MRI scan? I mean, I always thought the main reason was because he had a magnetic shunt put in after his first scan. So they did the scan, diagnosed him with normal pressure hydrocephalus, and then they put in a magnetic shunt in his brain. And so I was pretty sure the reason really they're not doing another scan is because he's being put between two giant magnets, which will literally like rip that magnet out of his head. Yeah, MRIs are no joke. They really want you to like make sure you don't have anything magnetic inside your body. Even like surgical implants and that kind of thing. If they're metal based, can be a real problem in MRI. But this answer is saying, so spatial resolution of the MRI is dependent on tissues with different water content. So if this guy still has normal pressure hydrocephalus, that's still going on, it's not going to show anything interesting. So if we go back to this scan, if you just keep doing MRIs, that all you're going to see is this big gray body, and you can't even sort of like differentiate structures underneath that. So scan is rendered useless. Yeah, that's pretty much what that question is asking for. Then there was, in the lab manual, so there was more stuff on functional magnetic resonance imaging, or fMRI. Yep, and then it's talking about, so the way that fMRI works is it differentiates blood content based on oxyhemoglobin versus deoxygenated hemoglobin because they have a different magnetic resonance. So more active regions of the brain will receive more blood, and also if you're having a stroke, you will see more. That region will seem more active on a functional or on an fMRI. Also, increased neural activity is detected as a different ratio of oxyhemoglobin to deoxyhemoglobin. So the amount of oxygen carried by hemoglobin changes the degree to which the hemoglobin flowing through the brain disturbs the magnetic field. Yeah, again, this is just talking a little bit about the physics of how an fMRI works. So what is the difference between an MRI and an fMRI? So MRI, I would say this is probably the main paragraph they need to know. So MRI differentiates structures based on water content of the tissue, and fMRI differentiates brain regions on the basis of the ratio of deoxygenated to oxygenated hemoglobin, because they have different magnetic resonance properties. As such, fMRI indicates areas of high metabolic activity, and also MRI imaging is not possible where patients have, like a magnetic device put into them. Yeah. So going to be plenty of examples in the subject where you have, what was I saying, where just like large blocks of content. So if you're the kind of person that just like needs to write everything down, I'm going to try my best to discourage that, because a lot of this is just taking like large excerpts from the lab manual. Again, like we can technically assess you on a lot of this stuff. But I'm just going to try to, as best as I can, point out where like you should be focusing your attention. In this case, I would say probably this bottom paragraph is the main thing we want you to take away from this, because it is directly phrased as like a question. This is certainly something that we can ask you. All right, we are almost finished. We can finish on something kind of cool. So intelligence was previously judged by the area of the human skull, or the size of the human skull. Why or why not is this misleading based on Einstein's brain? So I read a little bit about this. Einstein's brain was taken after he passed, somewhat unethically. The neuroscientists who sort of took it, I think, didn't strictly have like Einstein's permission, or there wasn't anything strictly saying that like, yes, you can take my brain and analyze it. But I think eventually later he got permission from his son or something. I don't know. It was a little bit, I don't think all nonsense is your favorite. Yeah. Particularly like, especially like around this time, there was just like, science was not like, it was pretty gruesome. We did a lot of dodgy things, but it was fun. All for the name of science. And the cool thing from this is that we dissected his brain, and we found that it was 200 grams lighter than the average brain. And everyone was like, what? How? He's like the smartest man in history. He's like historically thought of as the most like smartest person in the world. And so we were like, how could his brain be 200 grams lighter than the average person at the time? Does completely throw out this conventional wisdom that the size of a brain is what determines intelligence. But upon further sort of investigation, his parietal cortex was not divided by a deep groove. So normally there would be a sulci there, but the cortex was actually fused, which is incredibly unusual. And that fusion increased the connectivity between the brain regions that are associated with mathematical skills and visual processing, visual spatial processing. So this basically, we think, made him just a total whiz at maths, physics, all that good stuff. Whether or not that was something that he managed to do over a lifetime of like research and study, whether that was something that he sort of nurtured his brain to do, or whether it was something he was born with, is, you know, we'll never really know for sure. There's tons of cool research on intelligence and the way that humans who believe that intelligence can be impacted tend to be more intelligent, which is sort of a self-fulfilling prophecy. So in itself, it's a weird philosophical debate, versus people who believe that intelligence is something that's predetermined in birth tend to really struggle in terms of academia. So it's definitely an interesting topic, and something that, yeah, Einstein's brain was definitely something that has sparked interest in this as well. So because of that fusing of the areas, we believed that intelligence appears to be dependent on increased connectivity between neurons. So not necessarily how many neurons you have, but it's more about how well connected they are. So Einstein's brain had a higher density of glial cells, which are your like support cells. They also like nourish each neuron. So the relative number of support cells in specific brain regions may also be linked to intelligence. So those are kind of just the main lessons that we took away from Einstein's brain. So you're saying Einstein cheated. It's hard to say whether, like, we didn't have his brain when he was born, so we don't know if he was born with a fused parietal cortex. It's only really something that we can try and look at these days. But yeah, in an autopsy basically revealed that his brain was smaller. He had a small brain. You can say that about Einstein. He had a small brain. It's not very nice, but it's true. But he had increased connectivity between his neurons. So he was, yeah. Very interesting brain. Very interesting topic. Would intelligence relate to the thickness of the myelin sheaths of the neuron? So that's interesting. So the myelin sheath is what like increases connectivity between different neurons or along a nerve. Would intelligence relate to the thickness of it? I wouldn't say necessarily. I think this is saying increased connectivity. Yeah, you know what? I think you'd be right. I don't know. I think that's quite a contentious topic. It's not something that we would definitely know for sure whether thickness of the myelin sheath is related to connectivity, intelligence. But for the purposes of this subject, I would like to think that, yeah, thicker myelin sheath potentially would be related to more intelligence because it is increasing connectivity. It's increasing the speed at which a signal can pass between neuron and neuron. Yeah, cool question. Questions for revision. All right. Yes, this is homework. I'm sorry. So it's just a couple of questions that again, they're from the lab manual. So technically we can, yeah, sorry. Technically we can't ask you any of these, but I'm actually just wondering now how many of these would be from like the lecture and stuff. What are the 3 layers of the meninges? The first one, already covered that. You guys should knock that out of the park. Oh, by the way, I'm not gonna like mark this or anything. I'm not gonna like keep track that you guys are doing any of these questions or anything. It's all kind of up to you how much you want to put into this subject, how much you've got going on. Obviously, a lot of you are still going full time, so you can only dedicate a certain amount of like time to my tutorials and that kind of thing. But the revision stuff is there. The revision stuff is there. All these slides are there as well if you want to come back to them. This one is being recorded as well, as all of them will be, and going up on UTS Online. Is that everything? Yeah, it is. All right. Awesome. Good timing. Just before 4 o'clock. So I think I'm going to wrap it up there today, guys. What else will I just mention? Yeah, I always put my email up at the bottom of all these slides. So if at any point you don't, like if you're not watching this live and you just like pause the video at some point and you're like, oh, I really have a question that I want to ask Luke, just email me. Email's there. Should get back to you relatively quickly. Feel free to ask me anything about the slides. And yeah, let me just double check what we're doing next week. Next week we're doing, yeah, synaptic transmission. So carrying on kind of what David was doing. So it's a lot of stuff about concepts. You know, it's not going to be more of what we did today. This was just kind of a one-off. More membrane potentials, which is great. I remember doing them every semester for 3 years. I was so sick of action potentials. But yeah, we get to do just one more lecture on it. And then we get to do some cool stuff. Epilepsy, sleep, anxiety, depression, schizophrenia. That's what we're here for, right? All right, it's 5 to 4. Thanks for coming, guys. Thanks for coming to my birthday party.
Thanks, take it easy.
And everyone who's not here, that's watching this later, I'll be expecting your birthday presents my birthday presents in the mail.
Happy birthday, Luke. See ya.
Thanks, guys. See you guys next week.